Before we jump into the actual platform here, what we'll do is we'll get started with this slide about where to find help with FlowJo, just in case you run into any problems or have any questions. I highly recommend just emailing tech support@flowjo.com. That's the easiest way to get an answer back. Generally speaking, they'll get to you within the hour, if not by the end of the day. You can also try calling either the 800 number or the 541 number that is listed. We have manuals and help documents available on our flowjo.com website. If you simply navigate to flowjo.com, you'll see a tab that says Learn, and underneath the Learn tab, there'll be several options for webinars, tutorials, recorded webinars, etc. So there's a lot of information that is freely available to you, and you can certainly will find plenty of material to go through, and a lot of it is specific in that it targets specific components of the FlowJo application and what you're attempting to do. There are some new tools. I should mention SeqGeek, our single-cell RNA sequencing analysis package. You don't need to use R to find differentially expressed genes between populations or between samples. It's a pretty cool tool. We do have webinars for those for SeqGeek as well. Those are usually scheduled on Wednesdays. In any case, you can always check our webinar schedule to see what's coming up. We also have a new program called FlowJo Envoy, which helps you with program, basically project management. It handles your data, communication, and any of the sharing aspects of a normal LIMS system would have. The FlowJo Envoy system has a much more enhanced capability in that it keeps the context of your data alive so that it never gets lost, and it presents you with a really nice dashboard that allows you to visualize where you are on any given experiment. We also have some new plugins that are available on the FlowJo Exchange. I'm not going to go into those, but suffice to say, those are some pretty cool tools that we have available. All right, we'll jump right into FlowJo here. I should mention that most of the cell cycle type of experiments, we're staining our samples with DNA intercalating dye like DAPI or Hoechst. Also, propydium iodide and things of that nature are fairly common. It seems like every year there's a new DNA binding dye or new dyes for different applications. Just keep in mind, you want something that you can use with cells that are either fixed or fresh, and you have to pick the correct dye for the appropriate application. And of course, just be wary of the spillover of the dye, especially if you're attempting to use multiple colors for a cell cycle experiment. The one I have in front of us is a pretty simple experiment. If I actually just open up the graph window here, which is off to the right, you can see that from the drop-down menu, we only have DAPI and BRDU here that were being used in this particular experiment. So there's not too much that we have available. I went ahead and I've already gated on the cells in this experiment, and it's really the only other option that we would have after getting the cells together is maybe taking a look at the ploidy of the cells. So it's up to you if you want to create a gate around the euploid cells. In this case, the bulk population that you see here off towards the left is going to include the 2N cells here, and then the 4N cells are at the opposite end of this barbell-looking population, and in the middle is generally the S phase. Now, as I mentioned, you may want to create a gate around the euploid cells because the algorithm that we have doesn't incorporate the polyploid cells. In other words, you cannot model the polyploid cells. You can quantify how many of those there are, and so if I were you, a lot of times I like to leave the cells kind of untouched here, and I don't go ahead and gate them with a euploid gate. I leave them alone. So since we have only these dimensions here, I was only really able to clean up, the cells by getting rid of some of the debris at the bottom, and other than that, we have to go with the vast mix of cells that we have in this particular data set. I've already created the cells gate and copied it over to the rest of the samples in the group. Hopefully all of you are familiar with that operation. And then from here, what we're going to go ahead and do is we are going to go ahead and try to model these cells in where they are in a given stage in the cell cycle. So in order to invoke the cell cycle platform, the way to do it is go ahead and select the population that you wish to run the cell cycle node or the cell cycle platform on, and then go up to the Tools tab and click on the biology band. If your workspace is stretched out a little bit, a lot of times you will see that you don't actually have to click on the band. The option should be immediately available. So you click on the cell cycle button. It will introduce a node here that says cell cycle, and it should be indented underneath the population that you are targeting. Okay, so in my case, we've started with the first condition, and actually what I ought to do here is rather than initiate the platform on a sample that has some drug applied to it, it's wise to start with your control samples first. So I have a series of samples here labeled control. There are 3 of them. I'll go ahead and initiate the cell cycle platform on one of these controls here instead. Okay, and I'll explain why here in a moment. Okay, so for most cell cycle experiments, it's wise to have a control sample that is random cycling. Now, this is useful when you are looking at or using tumor lines, because a lot of tumor lines or cell lines that you have in culture, they're continuously growing, and it's easy to identify the G1 phase of the cell cycle and those that are in G2, or those that are replicating, have replicated their DNA and are ready to go through mitosis. They appear essentially as 2 peaks here. The higher peak is generally speaking the G1 population, and the smaller one is going to be the G2 population. In some instances where you're using primary cells, a lot of times you will not see much of an S phase, and you may not even see a G2 phase of the cell cycle. So in those cases where you're using primary cells and those primary cells are not cycling, it may be helpful to have a control that arrests the cells in G2. And drugs like nocatazole or colchicine do that pretty well. They're microtubule inhibitors, and they will lock your primary cells in G2 if they happen to be cycling at all. There's also, in the case that you cannot identify the G1 phase, there are drugs like acetylcholine and L-mymosine that inhibit DNA synthesis at the very beginning, and therefore will lock the cells kind of at the very, you'll see a very nice peak for the G1 stage of the cell cycle. So I would encourage you, if you are using primary cells, go ahead and use L-mymosine and colchicine for your G1 and G2 controls. If you're using cell lines, normally you can get away with simply a random cycling population of cells like we have in this case. Okay, there are 2 models to think about when you want to model the cell cycle. If you click on the model panel down here at the bottom, it'll expand, and that window will open up and show you 2 different model options. You have something called the Watson Pragmatic, and then you have another one called the DeanJet Fox. Okay? The difference between these models is simply how they arrive at the S phase, or how they calculate the S phase. Okay? The Watson essentially is considered a subtractive model. So what happens is, in the Watson model, we take what we calculate for the G1 phase, which is outlined here in the kind of the blue histogram. We add it to the amount of cells that are being found for the G2 phase, and then we just subtract the sum of these 2 from the total DNA, or the total numbers of events, essentially, that we have in this channel, the DNA channel. And then whatever we're left with essentially describes the S phase. Okay? The DeanJet Fox, on the other hand, uses a polynomial. So it actually attempts to model the S phase using a mathematical function rather than having it be a straight subtraction. So as a consequence, you'll normally see quite I would say, much larger swings in the S-phase quantities when you use the DeanJet Fox model. Both are good models, both are published, so it's really entirely up to you which model you would rather use for your analysis. I'm going to stick with Watson for this tutorial, but we may click, onto DeanJet Fox here off and on. But just for the sake of keeping everything, in tune and on the same line, we'll go ahead and just use the Watson for the time being. All of the constraints that I'm going to talk about in this webinar will apply to both models. So it's entirely up to you, again, which one you want to use, but the constraints and how you go about the constraints are going to be exactly the same. All of the statistics that are associated with each one of the phases of the cell cycle are indicated down here in this first panel just below the axis drop-down menu. On occasion, FlowJo may get the wrong DNA dye or the wrong channel listed. You can always change this by simply clicking on the drop-down menu and then selecting the appropriate channel. In this case, it's done a good job and it's found the DAPI channel, so we're okay. But on occasion, sometimes it'll put the forward scatter or some other channel that's unrelated. Just make sure that you change it over. The other thing to keep in mind here, again, about the statistics, there's something called the root mean squared here. I'll talk about this a little bit later. But the percentage of G1 cells that the model is currently finding is indicated here, roughly 32%. The percent S phase, which is outlined in this kind of yellowish color, indicates about 40% cells in S phase. And then, of course, we have the G2 peak over here in green. Percentage here is indicated almost 15%. Okay, the purple line that encompasses all of the histograms that you have within this model is essentially the model sum. So this is essentially what FlowJo is using to calculate and model the sample. The actual raw data here is outlined by the black line. That's the amount of cells and the number of events that you actually have in the experiment. So the gray and the black line correspond to the raw data. The colored histograms here and the colored line reflect what the model is doing with your raw data. Now there are some other statistics here. The G1 mean, this is just telling you where the peak value or the modal number is for the G1 histogram. In this case, it's about 193 units. So if you got down here and you actually zoomed in on this, you might see that right around 190, 193, that's where that modal number is going to pop up. In the case of G2, it's roughly twice that at 380, 385. And then we have two other statistics here. We have the G1 CV and the G2 CV. The CV is essentially telling you the width of the histogram or the coefficient of variance. That's what CV stands for. So it's essentially the spread in the data. And I'll talk about these two statistics here in a moment. Now the other two stats, the sub G1 percentage, roughly 5% here, and that which is greater than G2. Again, these are the greater than G2. These are the polyploid cells that we don't model with Gaussian curves or anything, but they are quantified here for you so that you know that outside the model on the right-hand side, there's roughly 9% of events that are hanging out here, and then sub G1, there are roughly 5% of events hanging out on this direction. Okay, I'll discuss what the cell cycle gates in the workspace do in a moment. If you want to play around with the model and essentially the different components here, if you just toggle these boxes, you'll see that the model sum is that purple line that I discussed earlier. If you can toggle that on and off, it'll get rid of it. Also the components here, if you don't want to see them, I don't know why you would do that, but in any case, you would go ahead and untick that box. And of course, if you want to unfill and just look at them as sort of wireframe histograms, you can do that by simply toggling that fill components box. Okay, now one of the first things to think about when it comes to the cell cycle, again, we're going to base all of our observations off of a good control. So what we want to do here is we want to fix the range for the G1 peak, essentially where we would expect the G1 population of cells to fall. Now granted, if you are using the same cell line for all of your experiments, you can keep this gate relatively narrow. If you're using multiple cell lines, you probably want to initiate separate cell cycle models for different cell lines that you have in the workspace. Okay, in our case here, I might set this up to be a fairly narrow range here. The minute you click and drag across the interface, it applies this small drop-down menu, which allows you to either constrain the G1 range or the G2 range. In this case, we're doing it across the G1, the first set of cells here off on the left-hand side. So I'll go ahead and say constrain G1. Okay, if you think that's a little narrow, you can always widen this or taper it however you prefer. I'll extend it off to the left a little bit. Sometimes your populations will move a little bit as a result of treatment. The other constraint we're going to set here right off the bat is the G2 constraint. Again, because this is a random cycling population of cells that are grown in culture, it's pretty easy to see the G2 area. So we'll go ahead and click and drag across the G2 region, and we'll say constrain G2 range. Okay, once you have these ranges constrained, you at least have what I like to call the goalposts for the rest of the experiment. Okay, so the model then is going to look for peak values that fall into these ranges, and it's going to attempt to model the S phase accordingly, somewhere between these two goalposts. Okay, so once you have the G1 and the G2 phases or G2 peaks set up, the next thing to consider is the width or the CV of the histogram that you are displaying here in the model. Okay, in our case, the G1 constraint here for the peak is a range that we defined by dragging and dropping, or just drawing a line across the interface here, and that's what's described in the two boxes down here. But the CV has a button, and within that button are several options. Okay, you can leave the CV unconstrained, that is, you let the model determine what the CV should be from sample to sample. Or you can set the CV to a particular value, which is indicated by the equals n. And then, of course, if you want the CV to be the same as the other peak, in this case the G2 CV, you can go ahead and say equals G2 CV. Now I'm going to talk a little bit about the CV and why we should set the constraints the way we're going to set them. Okay, the first one is really more or less a matter of eyeballing the model that we have in front of us. Okay, so when I start, I look at the G1 population here, and I kind of play around a little bit with the CV associated with the G1 peak. In this case, it looks like it fits that first histogram pretty well, or that first spike in the data, if you will. But we can play around with this and try to increase the CV just a little bit, make it a little bit fatter to kind of fit this histogram a little bit better. As we can see here, the G1 CV is currently set as calculated as being 7%. What we can do here is we can go ahead and just say equals n, and I'm going to fatten this up a bit. Instead of having it be around 7, what we're going to do is maybe fatten that up to about 10 and just see what happens when we increase that width. Okay, see how the histogram is a little bit more tapered towards the bottom here and kind of a little bit fatter overall with respect to the model. Okay, now if we go back to, just to give you a quick reference here, we'll go back to 7. You have to kind of click off that box in order to get it to apply. But the minute you apply it, the model will reflect what you have there. Okay, in my case, between 7 and 10, as far as personal taste goes, I would say that the 10 is a little bit better of a fit to my eye, simply because it looks better. Okay, but there's another statistic here in this panel at the top that helps guide some of our manipulations in the model. And this statistic is called the root mean squared statistic. And what this statistic does is it tells you essentially the distance between the model that you're calculating here, so essentially the purple line, and raw data, which happens to be outlined with the black line. The smaller this number is, it means the tighter this model fits the actual raw data. Now, generally speaking, if you have an RMS, a root mean squared value less than 10, you're doing pretty well. In our case, you know, we're well under 10, and a lot of times this, you know, this statistic is also going to be influenced by the numbers of events that you have in the model. So I would generally say, you know, start out with no less than 10,000 cells acquired, and within the model you should have, you know, a few thousand cells at least. So I'd say anything greater than 1,000 cells, you'll normally see values here that are workable. Okay, in this case, if we come back here and we look at the changes that we make for the CV, let's see how it influences the root mean squared value. Okay, so if I set this to 10, okay, click off of this, you'll notice that the RMS value increases a little bit, meaning that the distance between the raw data and what my model is doing has increased a little bit. So it means that this is not fitting, you know, the model that I'm generating here is not fitting quite as well as how flow. And you can kind of step this back a little bit one by one, you know, to see if some of these minor changes are going to improve the model significantly. And generally speaking, if you're within, I don't know, 0.5 units, I would say that, you know, at that point you're talking pretty minimal amounts, and you shouldn't be too concerned with how far away you are from the from the model's previous setting. In this case, if we set the RMS, or sorry, the G1 CV here to 8, you can see we get an RMS of about 3.4. If we go back to 7, right, we start to decrease it just ever so slightly. So whether you keep it at 8 or you keep it at 7, at this point it's really a matter of semantics. And I would say that a little bit wider of a CV here, at least to my eye, is a little bit more pleasing. And so I'm going to keep it there at an RMS of 3.4 and the CV at 8. Now, as far as the G2 CV is concerned, the CV, the reason that we have this spread at all, this is actually a property that is intrinsic to the cytometers themselves. It's actually related to the error of the photon counting error of the cytometer. So, in principle, we would expect that if we stain our cells to saturation, which we normally do when we use a DNA intercalating dye, we generally have about 1,000 times more dye molecules than DNA molecules. We would expect in that case, when we saturate our staining, and if our cells happen to come from a clonal population or from the same species, right, we would expect that the DNA from cell to cell is exactly the same number of chromosomes, the same length essentially. We would expect that, there, instead of being a histogram here, there would be just a single column. Okay? The reason there is not a single column here and that there's actual spreading going on is because because the cytometer has photon counting error. Just like every other instrument that we use in the lab, there's an error rate, in this case, roughly around 8%. Okay, 7 or 8% in terms of the photon counting error. Now, if this is the error, the spreading is associated with the error of the machine, we would expect that the error that you see here is exactly the same as the error that you would see on this side. In other words, the type of cells or the fact that cells are in G2 should not influence the error of the machine. The error of the machine comes from the machine itself. Okay, so in this case, all I'm going to do then is take the error that I see here, which happens to be 8%, the CV there, and I'm going to tell it to go ahead and be the same in the G2 population. Okay, so I can either constrain this by, again, saying equals n, or I can kind of use the shortcut here and just say equals the G1 CV. Okay, when I set that to the G1 CV, both of these guys should appear the same here in the panel at the top. Okay, and now I know that my peaks, my peak values here are constrained to these positions, and the CVs are going to be about 8%. The error in both instances should be the same. Okay, so at this point, this is where I would go ahead and take the samples that I have in the workspace. Okay, and the way to do this is to go ahead and drag and drop that node onto the cells node that you have in the groups pane. Okay, so you have to take it up here, try not to miss, get it on the cells node in this case, such that all the other samples will inherit it in the appropriate location. Okay, once that's been done, if you go back to the platform menu, you can now make use of the green arrows going right or left. So if we click to the right, right, you can see here we have some really ugly looking samples. Right? In this case, this is another control, but we have a number of events that are on either side of the histograms here. But because we constrained our model to that first sample, right, we can be confident about the results as they're coming down from after being applied to other samples. Okay, so we'll go here, check this out. Right, this is a really interesting sample where we've actually treated the cells with some. The cells were treated with some sort of S-phase blocking inhibitor. In this case, it most likely was acetylcholine. I'm not familiar with the details of the experiment, but the cells were locked in. The cells were locked in G1 for quite some time, and they actually began slipping into S-phase, and then they were released from that drug into regular medium. And you can see here that we have kind of a very broad shoulder off to the right of this major peak. And it seems that FlowJo is calculating this lowest bin here as the G1 value for that particular sample. Okay, if we advance another step, you can kind of see the same behavior occurring here. Right, we have this sort of broad shoulder off towards the right. Right, and so on and so forth as we go through the data set. Okay, now if you want to modify the peak here so that it more or less helps to reflect, a little bit more where it appears in these other samples, you can always move that G1 range. Okay, but I caution against doing this simply because we had set these up on controls. Okay, but a lot of times this might help if you actually make it a little bit narrower. Right, go ahead and change that model. Okay, if you want it to update to what you're seeing here. I don't recommend that you do this. I'm just showing that you can. Okay, go back to the workspace. If that's the new position that you like, go back to the workspace before you lose what's going on there, and go ahead and select that cell cycle node and repeat the process. Drag and drop this up to the cells node. Go ahead and say OK. And then it will copy everything across to all the other samples. And now we can see what happens. What is the effect of narrowing that range on the rest of our samples? Again, here it appears that the peak is pushed off towards the left. It's not going to that high value that we have here off towards the right in the shaded area. Okay, and how does it compare, more importantly, with our controls? You see how our control peak here is a little bit off towards the left. This is an interesting, it's an interesting data set in that sense that the peak here is okay, but in some of the other some of the other treated samples, the model actually prefers the bin towards the right. Okay, this actually looks okay. So with respect to the controls, making that modification, I return here to, this is control number 1. Again, I'm missing part of the peak here off towards the right-hand side, so I might go back and expand that. I think this is a better judge of the position of that particular G1 gate. So again, once we make that change within a model, especially on a control, go ahead and copy that to the rest of the group. Okay, in this case I missed. So again, just repeat the process until you nail it. Just say yes, and it'll go ahead and update. Okay, and that will apply that model to all the rest of the samples. Once you have these data, at this point you can you can play around with the models if you'd like. So once you have the gates and constraints in place, sometimes it's a matter of just visualizing which model you prefer. So you can click on the DeanJet Fox. If you tick this box down here where it says with synchronized peak, what this will do is it actually splits up your S phase in half so that you have two S phases, the front end of the S phase and the back end of the S phase. I'm not exactly sure why this is useful, but that's essentially what the algorithm does. Okay, and then from here, once you've kind of visualized the different application of the models across all the other samples, again, it's a matter of personal taste, which one you want. Just pick a model and stick to it, right? In my case, again, I changed the model, but I did not apply it to the rest of the samples. I'm kind of curious how it applies to some of those that are kind of erratic with their distribution. You see how the S phase here has that kind of nice round bell-shaped looking curve? Again, this is due to the DeanJet Fox having to use a polynomial function to model the S phase rather than a straight subtraction. Okay, if you go back to Watson here, you'll notice how it's very scratchy, kind of this rough interface, if you will, whereas DeanJet Fox will try and smooth that out for you. Okay, definitely a little bit, to me it's a little more appealing to the eye, but whether or not I would use DeanJet Fox, I don't know. I used to use Watson all the time, and it's just because of what I started with. But as I mentioned before, either one will work. Okay, from here, you will see that there are a couple of options. There's an option here in the gates box where it will say create cell cycle gates in the workspace. So if we tick this box, what it will do is it'll go ahead and generate gates based on the curves that you have here in the model. If you want to actually visualize these gates, you can either double click on these. You can see the histogram here. If you click the up arrow, you'll actually see the ranged gates that FlowJo is using based on the model. Now, it's important to remember that these gates that are produced here, positionally, they reflect what you see in the model, right? So the gate range, the area that you're finding here in the model is actually reflected in the graph windows, okay? But the quantities that are contained within each of these gates are not going to match what you have in the model here. Again, the model is working with a probability of cells being in one population versus the other. It's assuming essentially a Gaussian distribution of events for the G1 and G2 phases, and I mentioned earlier whether it's a subtraction or a polynomial with respect to S phase. So don't expect these gates, right, to reflect the percentages that you see over here. These are solely for the purpose of visualizing and creating overlays in the workspace. So if you want to overlay these with each other or with other samples, you go ahead and open up the layout editor and then drag in the remaining gates, and you can drop them on top of one another to see what the model looks like. Now, by default, FlowJo smooths these histograms when you overlay them, and so when you look at the graph here, it appears that there is overlap between these different histograms. If you double-click on the graph itself, you'll notice that on the Specify tab here, there's this option where it shows smoothing. If you remove the smoothing and apply it, you'll see that those gates are they're really just hard tapered gates, and the cells that are contained within each one of these gates are not overlapping. So unlike what you see in the model. Okay, so if we go back to the cell cycle model here, you can simply drag and drop the cell cycle node directly into the layout editor. right? And if you want, you can batch this across all of your samples. I'm going to delete the overlay that we just created for a second here, right? And the model will come with the visualization that you kind of see in the platform window, and then a pane off to the right that shows you all the statistics that are contained within the platform window itself. If you don't want that stat pane, you can always select it and then delete it. There are other manipulations that you can make here in the graph window itself. The plot, if you double click, you can change the fonts, of course, the legend that's associated with it or not if you want it, and then any of the annotations that are associated with the graph. Once here, if you want to batch this, you can across all samples. So I'm going to put this in columns going down so it's a little bit easier to see, and I'm going to get rid of the show page breaks for the time being, just so that we can easily see the graph. Okay, once you have it here, you can go ahead and go to the Layout Editor tab here, click the Create Batch Report, and this will create a tiled report that shows you all of the graphs that you have, right, for each one of the samples in the current data set. And it will show you essentially what we have in the model. Okay, in addition to the layout editor here, you can take the cell cycle node, and you can export those statistics directly to Excel or create a CSV if you click on the table editor tab at the top. Okay, so the table editor is the module that we use to export our statistical functions. Okay, so click on that, go ahead and click and drag the cell cycle node into the table editor, and it will populate here with all the statistics that are associated with the platform. So everything that you see here is going to be reflected in this table. Okay, if there are any statistics that you don't want, perhaps you're not really interested in seeing what the G2 means are and the G1 mean, you can go ahead and get rid of those, as well as the CVs and some of the other stats that may not be so super useful for understanding what's going on with a given experiment. We'll get rid of the synced populations as well. Okay, once you've got this down, we can go ahead and make the table look slick if you want. Go ahead and shift click to highlight all of the samples or all of the statistics. And then if you head over to the Visualize tab, you can go ahead and click this heat map option, which will put a little fireball next to your stat. And now when you click the cog button here off towards the right, or if you navigate back to the table editor and you say create table, you'll get a table here that has heat mapped values for each one of your statistics. Okay, now in this case, I'm not exactly sure why the RMSD is not popping up, but all the other statistics associated with the cell cycle model are presented. Okay, you can copy and paste this if you want directly to Excel. You can also copy and paste it into Prism, I believe. I haven't tried that in a while, but it used to work just fine. The other alternative here is to go ahead and change this from display if you want to create an Excel file or a CSV. Right? Just click this button, say to file, okay, and then when you do that, it'll give you the option here to save it as a CSV or an Excel file. There are some other options here as well, but by far these two at the top, or three at the top, I should say, are the most popular. Okay, when you select the file type, just make sure that you give it a destination, know where it's going. A lot of times sort of dumps the file kind of into the ether of your hard drive. So you just want to make sure that it's going to someplace that you recognize. In my case, I might just dump it on the desktop. Click Save, and then remember to click that Create Table button, and it'll actually produce that table in your desktop. Okay, from here, saving your work is simple. Just click on the File tab and say Save As or Save. If you click Save, it'll save it with a timestamp name. If you click Save As, it'll give you the option to save it as whatever name file you would like. Okay, with that, I will open up the forum to questions. I saw that there was one question that popped up during the tutorial. And I'll try to answer that question right now. And if you guys have any questions, you can certainly open up your mics or go ahead and use the chat box, either way. Okay, so there's a question here that says, why doesn't the sum of the percentage G1, S, and G2 phases result in 100% in the Watson model you showed? Okay, a lot of times you have to remember that this is a computer model, and sometimes the algorithms that, that are being used here cannot make full use of the data that are in front of them. Now, it's hard to explain in its entirety, but the model is constrained to a certain extent. And when we constrain the model, a lot of times you can think of constraining the model as borrowing some of the total data that we have available to us and applying it to those constraints in the model. So when we do that, we sort of remove some of those data points, or we remove some of that data from being able to be calculated in the model in its entirety. So a lot of times this may fall short of the 100% mark. In this case, you can see here we're looking at DeanJet Fox, right? We have a G1 at 15, right? An S phase at 50, so we're 65%, and then we're adding another 10%, so only 75% only 75% of events here are being calculated for the G1, S, and G2 phases. Now the remaining cells are kind of hanging out here as sub G1 or greater than G2. So that the model doesn't equal 100% is not critical. It's not a problem. It's just a matter of the constraints that have been applied to the model. So in some instances, you'll see that it gets closer to 100%. Other times, you'll see that it falls well short of 100%. And not to be alarmed, it's just that's how the algorithm works, and there's nothing we can do to really increase or decrease that without having changed the model in its entirety. And so, for example, here again, we're falling short of that 100% easily within the first three phases. So I hope that answers your question. I know it's not the clearest answer, but if you want, you can send me your email, and I'll try to get you a more formalized technical answer from the engineers at headquarters. Okay, so there's a question here about the RMS. The bigger the RMS, the better then? No, the lower the RMS, the better the model, or the better the model fits the raw data. When you're moving when you're working with the RMS and there's very small changes to the model, at that point, whether it's bigger or smaller is kind of irrelevant. What you want to make sure is that your RMS is lower than 10. And if you have a model where the constraint that you have leads you to an RMS of 6, let's say, and then a different constraint leads you to have an RMS of 3, in that case, you could say, the RMS difference is large enough that you would go with the one that is smaller. But 0.1 and 0.2 differences here in the model RMS are relatively insignificant. Yeah, so if you're under a value of 10, it's pretty good. But again, the RMS, I'm giving you that as a benchmark because it's not going to be the same from model to model or from experiment to experiment. It really depends on how many events you've collected in your experiment and how many are actually going into the model itself. So for example, when we're looking at the cells population here that I have for condition A, we have roughly 11,000 cells being fed into the model. Anytime you have 10,000 or more events in the model, you're going to see that it's pretty smooth, and that RMS value is generally going to be fairly low. If I was to increase this to 50,000 events, you would see that the RMS score would probably drop to about 5 or less. So it not only follows the constraints and how good the model fits, it also is proportional to the number of events that you have in a given experiment. So just just keep in mind this is a relative measure, and it's specific to the experiment that you're working on. You shouldn't take this RMS and say, oh, you know, my RMS in this experiment is way better than the RMS that I had in another experiment, because you may have more events occurring in that second experiment than the first. Okay, so this is just meant as a guide here to tell you how well your model is fitting the raw data. Try to keep it low, you know, as low as possible, and obviously, to some extent, the model has to match what you're, you know, what you're seeing with your eye. Okay, are there any other questions? Okay, if not, I encourage you all to check out the Cell Cycle platform. Just play around with it a little bit. Just remember, you know, once you have the model in place, go ahead and apply it to the rest of your samples. Use your controls to set the constraints here in the lower section. Okay, set your G1 and your G2 on your random cycling population. Then go ahead and set your G1 CV, and I would constrain your G2 CV to be equal to the G1 CV. And that way we have a very nice tight model that reflects the intrinsic error of the machine as well as what's going on with the biology of the system. Okay, there's another question here about asking how to determine the gait for G1 and G2. Generally speaking, when we look at cells along the DNA parameter here, let me go ahead and, the width, not the peak, okay, the width of the histogram, or the G1 histogram. This is really more or less a matter of using your controls, and, you know, it's kind of a combination of personal taste and the RMS value here. So when I was constraining the G1 phase on this model, right, I constrained the region, and then I was looking to see how well my histogram, my blue histogram here, kind of meets up with the histogram that is outlined in black. Personally, I like it to fit a little bit, you know, a little bit wider so that it occupies the vast majority of space here. But as we saw when I played around with this CV a little bit, the better value was slightly smaller. So when we had this as a narrower CV here, we set this to 7. Right? If I click off of this, that RMS goes down to 3.9. Okay, so you could say technically it's better to leave that as a little bit, a little narrower of a histogram, because the model actually has a better fit here when I reduce it to 7. Okay. Let's see what happens when we reduce it to 5. Do we increase or decrease that RMS? Okay, and in the case of 0.5, we've gone, or 5 CV, we've gone way too far, right? We've bumped this up to 7. Right? If we're here at 6, maybe we'll improve it a little bit, right? From 5 to 6 is a huge jump, right? We've gone 2 points down, which is nice. And then again, as I said, it's really more or less personal taste here. If we're at 7, we get 3.9. If we are at 8, right, we go up 0.1 units. So the change in the RMS is not that huge between 7 and 8, right? But you are correct. You know, we could say that technically the 7 is a better number because this RMS is minimal when I set it to that value. Okay, but again, it's at that point where the variance is very tiny when I make these changes, it's more or less a matter of personal taste. All right, I missed the first 5 minutes, so I don't know what the control is. The control sample in this case is just a simple random cycling population of cells that are in culture. So I don't remember what kind of cells these were. I'm guessing they were HeLa cells, but it could have been HEC293s. Okay. To Ling Zheng, who asks, that means use the minimum RMS is better? Yes, go ahead and use the minimum RMS. That's a safe bet to go with. Okay, let's see here. We've got another question in the chat box. Sometimes it looks like the treatments move the G1 peak to the right, but when you apply the model, it belongs to the S phase. Is that right, or should we adjust the G1 phase in each treatment differently? Okay, so this is a great question, right? So I'll repeat it here. Sometimes it looks like the treatments move the G1 peak to the right, but when you apply the model, it belongs to the S phase. Is that right, or should we adjust the G1 phase in each treatment? Okay, so the problem with moving the G1 phase for each treatment is that the model is no longer based on your control, right? So you want, in order to be able to draw a conclusion and say, my S phase in sample number 1 is greater than it is in sample number 2, or whatever, right? We have to have some basis for that comparison. And the basis that we're setting up in this case is by setting it on the control sample. So even though it looks a little odd when we move to a couple of these, you know, ugly looking samples, right? I'm safe in knowing that in this case, the G1 and the G2 peaks here, I've constrained these to where the controls are, okay? Not to what my eye sees with respect to what's going on here, right? You don't want to change these values on the treated samples because these values are the constraints that determine the ranges using the appropriate controls. Okay? In fact, if anything, I've probably been a little bit generous here by making these gates relatively wide, and I could have been a little bit more narrow with respect to their positions. Okay, so just remember, once you constrain the model, you should be constraining the model using your controls. Once those constraints are put in place, no matter how weird it looks on your experimental samples, don't worry about it, okay? Because you have the confidence that you created those gates, those constraints on the control sample. If you change this around for each one of your experimental samples, right, we can no longer compare the results that we get from sample number 1 to sample number 2 because we're kind of picking and choosing where we think that G1 peak ought to be. Okay, any other questions? I'm searching both the Q&A panel and the chat box. Either one is good. Okay, so I'll give you guys a little bit of time here. The other thing too is that this webinar is being recorded. So if you missed an earlier section, you can always come back to flojo.com and you'll see our recorded webinars, and you can simply download it and you know, review it if you would like. Okay, I'll wait another minute. If there aren't any more questions, I'll bid you all a good day, and you know, enjoy the Christmas season coming up. We have Christmas next week and New Year's the following. So tech support should still be available in case you guys are working, but highly encourage you, you know, take some time off at the end of the year, relax a little bit. There's always more experiments to be run in the lab. All right, well, I don't see any more questions, and thank you all for attending. If you have any that you can think of, you can always email me as well. So my email is simply jack@flojo.com. But again, the quickest way to get a response back is to simply send an email to techsupport@flojo.com. All right, well, with that, I'm going to go ahead and end the webinar. Thank you again for attending, and look forward to seeing you in the future.